Thank you. And thank you all so much for allowing me to be here. I'm really excited to talk to you guys, not only about my work at Baylor, but also my journey through college and to a PhD program, because I know most of you are kind of at that point where you're starting to look into colleges and figure out what am I going to do, which is a really daunting question. I also want to comment that this place is amazing, and the experience you guys get, I would have loved to have had at your age. So this is just so impressive, and I know all of you are just as impressive. So as my title says, I'm going to the type of research I'm going to talk about after I talk about my journey to grad school is going to be about this blue-green algae issue and the cyanotoxins that cause that. But if you were to ask me who I am or what I do kind of outside of this setting, I would tell you that I'm a limnologist. So limnology isn't exactly the most common word used, but I want it to be. I want everyone to know what a limnologist is. So limnology is the study of inland waters. So pretty much anything that is not the ocean. So we're talking streams, rivers, and what I really focus on, which are lakes and reservoirs. Reservoirs are just man-made lakes. And right here I have a couple pictures of the beautiful lakes that I've gotten to work on. I love this being my office. I think it's the best job I could have. And one other thing about limnology is no matter what you're interested in, I bet there's a place in limnology for you. So limnology in itself integrates many, many different fields. Physics is an important part of limnology. So when you think about rivers flowing water into the lakes, we have a term called water residency time, which is how long does one single drop of water stay in a lake? And then one single drop of water stay in really any body of water, but I focus on lakes. And physics is a really important part of that because of flow. And there's a lot of awesome mathematical formulas that we can use for this. Chemistry, water chemistry like pH. pH balance is extremely important whenever we're thinking about the aquatic organisms that live in the water. You wouldn't want to be swimming around in bleach, right? You don't want it to be too basic, too acidic. They like it at a pH around 7. Geology is also a huge component of limnology. If you have seen limestone before, limestone is actually calcium carbonate, and a lot of lakes, especially in this area, are encompassed in limestone rock, which can dissociate into the water, and that can cause an overabundance of calcium and also some carbon issues. And my favorite part of limnology is biology. So all the living stuff going on inside these water bodies. And I am biased towards biology, but whenever any kind of limnological issue comes up, all of these work together to try to solve the problem or figure out what might be going on. So you really can't have one without all the others. But to get here, to be a limnologist, was not a straight path. I never expected to really go to graduate school. Being a PhD student was never on my mind when I was in high school for sure, let alone a field I had never heard of. So originally, I'm from Oklahoma. I'm from Edmond, Oklahoma, which is a small suburb above Oklahoma City, so where I have that star. And when I was in high school, I didn't even know if I wanted to go to college, let alone what I might want to be. I know in high school we have a big push, like go to college, and I 100% after going through it support that, but I just didn't know what I wanted to do. I went to a public school where there wasn't a lot of guidance for that kind of stuff, and our advisors didn't really talk to us about college too much. So I was being told, just go. This is something you do. And I decided to apply out of state to a school, St. Louis University, which is a really good private university. And somehow they accepted me. I'm not really sure how that happened. I wasn't exactly an A student, but I got in. Well, when I got in, they asked, what's your major going to be? And that was something that I didn't even know you had to, say. I knew nothing about the college process at this point. And I, kind of just a gut reaction said biology, which is a little ironic because in my biology course in high school, I was not doing well. I was barely making a C. And an instructor actually told me I should not go into biology. I would not do well in it. Don't do it to yourself if you're ever interested in this. So I think more the sarcastic part of me, when asked that by my advisor, my gut responded, biology, why not? Well, so there I was, enrolled as a freshman in a new city, a new state with this major. And the way St. Louis did it was the dorms you were in, they put you together with people with similar majors. So almost everybody who went into biology wanted to be a doctor. So everybody I was surrounded by in my dorm was pre-med. So I kind of thought, cool, I should totally be a doctor. That sounds great, a medical doctor. That sounds super prestigious. People love it when I say I'm going to be a doctor. Why not? Well, life definitely happened. and I realized that I was not well prepared for these courses from high school. The introductory biology course I took, barely got a C. Introductory chemistry class I took, I ended up dropping it because I was not doing well, and I took a remedial course for no credit that I still had to pay for in hopes that I could learn this material that I just didn't get in my high school. And the tuition, because of this, the small scholarship they offered me, I lost because I wasn't keeping my grades up. And I could no longer afford to go there because the tuition was way too high being out of state and it being a private school. So all these things happened, and at that time I felt like it was, the end of the world. Well, that took me back to Oklahoma. Luckily for me, in Edmond, Oklahoma, a really awesome school exists called the University of Central Oklahoma. I'm sure you've all heard of Oklahoma State and Oklahoma University. Those are the two big ones because of football. UCO falls right under them. The sports aren't there, that's okay, but the teaching and the experience you would get, I believe everything happened for a reason because that school, top notch. So I was there, and luckily I had friends from high school who also went there, so I had someone to live with and dorm with. But I didn't change my major. I stayed a biology major, and I thought, you know what, okay, I should still totally be a doctor. I'll still keep taking human anatomy classes and things like that. But because UCO was more a teaching school, I was doing better in my classes. I no longer had to wait in a huge line outside my professor's office to get help. The TAs and SI people were really available for me, so my grades were doing better. And I would encourage you all to really think about that whenever you are applying to schools, is what school really fits what you want out of it. Everybody is different and every school is different. So, I stayed on that track thinking, yeah, okay, pre-med, this is good, everything's going all right. But then I took a cell biology class, and I wasn't doing terrible, but because my grades were doing much better, I kind of wanted to keep with around that B range. And in that class, I was doing around a C mid-semester, so I dropped it. But I had this idea in my head, I'm going to graduate in 4 years. I'm not going to let, the kind of downfall of that first semester affect when I hope to get my bachelor's degree. So I was taking a lot of summer classes, and it's like, okay, well, I don't want to put another summer class on top of what I already planned to take because I dropped this class. What else is available? So this was probably the end of my sophomore year in college. I was kind of just looking around at all the course things in the course catalog, and I found that a professor was doing a 2-week field course to Belize. And that sounded amazing. Okay, great, I can get a vacation and I can still get credit, wonderful. So I called him, actually. I called his office and I asked, I was like, I know, the deadline's coming up to apply for this. Do you have a spot? Lucky me, somebody backed out, he did. So I was able to enroll in this course, and because this happened at the end of the semester, the rest of the semester we spent meeting once a week, about 8 of us, talking about the plants of Belize, talking about the animals of Belize, talking about ecotourism and ecology and all these things that I had never been exposed to before, and it made me realize how amazing that whole side of biology really is. Biology can go beyond medicine, and I had never really been told that or was exposed to it. So I went to Belize, and we camped in the jungle, and we canoed up a river, and it was so life-changing to me that when I came back, I immediately asked that professor, how do I do more classes like this? This is interesting. This is really making me excited to go to class, which is important. You have to love what you're doing. And he told me about, plant biology and things like this, and I'm sure to a lot of people sound a little boring, but after this experience, I just wanted to know more. He also introduced me to a couple professors, and I started doing undergraduate research. I did a couple different projects. I did one where I grew this bacteria, Actinomycetes, to see how it could remediate chloridane, which was an agricultural pollutant that was still in our soil and actually has been found to be a carcinogen. And then another experiment that I did was looking at how lavender can prevent nematodes from affecting soybean plants. So just all this cool stuff going on that if I hadn't have done this, I would have never been exposed to. And I really wouldn't have changed kind of the trajectory of my career. So when I was working with these professors and I was going up on my senior year and I was about to graduate, I was talking to them and I said, what are my options really? What should I, asking, what should I do? I don't know what's next now that I'm gonna get this bachelor's degree. I also had a couple minors. I got a minor in chemistry and a minor in business administration. So my head was almost kind of going environmental management, environmental consultant type job. And one of my professors recommended graduate school. And he told me, he's like, well, there's plenty of options for a biology degree, but if you want to be a team lead, if you want to kind of be in that management position, getting your master's can only help you. So, okay. So I applied to Oklahoma State University, and I was enrolled in their environmental science master's program. Once again, I didn't really do my research. So if you walk away from nothing more here, please do your research before you apply to programs. They accepted me without an advisor, and that's not typical in grad school, at least in my field. In my field, usually you talk with a professor beforehand, and they accept you as their student, and they say, yes, you can work in my lab. I will be your mentor. They accepted me without that, which I didn't realize was abnormal. They were a newer program at the university, so they just didn't really have all their ducks in the row yet. So I quit my job I had, I broke my lease, I moved to Stillwater, where OSU is, luckily only about an hour away. I got very lucky with that, but still moved there, slept on some friends' couches trying to figure everything out, got a roommate, all this good stuff. Well, when I went to talk to the director of this program, he told me I had about a month to find an advisor or else I'm out of the program. Okay, what? And that kind of shocked me. So I pretty frantically started seeing what research is being done at OSU, who needs a student. And once again, very luckily for me, Dr. Andy Jalowski at Oklahoma State University was looking for a master's student. And that's when he introduced this beautiful word to me of limnology, because I would have taken thing, which I also don't recommend. But he explained the work he did to me. He said, okay, he's like, I'm interested in looking at how the watershed of lakes, how the land cover of that watershed affects what nutrient or element is limiting the growth of algae. Are you interested? And I was like, yeah, I am definitely interested. That sounds cool. I can be outside. I can, be hands-on. Sure thing. So throughout my master's process, I definitely gained a love for limnology, but I also gained a love for teaching, which is another thing I was never exposed to. But as a graduate student, at least in the sciences, usually your stipend, what you're paid on, is based off of teaching. So if you are in someone's lab who has a grant, they can pay you off that grant and you're called a research assistant, or you can teach for the department, their labs, and then you're a teaching assistant. So during my time here, I was a teaching assistant for the whole 2 years, and I loved it. And I realized how I want water science in more classrooms. I want it in high school classrooms. I want it in undergraduate education. I want water science everywhere. So that really sparked my love of both of these things. So during my time there, like I mentioned, this is just kind of a brief graph of where my master's took me. So I lived in Oklahoma. I moved there when I was about 14 with my parents. And for my project, Oklahoma was a really awesome thing for this whole land cover, because when you are in the eastern part of the state, or I'm sorry, the western part of the state, it's very deserty, probably just like Texas, very dry. You go to the other side, it's extremely forested. So I had a wonderful gradient of very, very much forested land cover to very much agricultural and desolate and everywhere in between. So I picked 25 lakes from around the state, and I got to drive to all of these lakes. So I also got to see my state in a very different way. And what I would do is I would collect water from these lakes and I would perform bioassays. So what you see here are 3 different lakes, each shelf representing water from a different lake. And I would spike them with nitrogen, phosphorus, iron, and then all possible combinations of them, as well as a control which didn't have anything. And pretty much I would track their growth, the algae's growth, and whatever grew more, those were the nutrients it was limited by. So then I was able to kind of point those out on a map, and I don't have the data here for this to show, but it really correlated to land cover to kind of say, okay, if your lake is in a watershed that's 70% forested, this is probably the nutrient it's limited by, so this is the nutrient you should control for, because if you control for it and it doesn't enter the lake system, we can't have a bloom. And so that was a really awesome project. With this after, I thought, well, I love teaching and I also love doing research. Being a professor sounds like a perfect job for that. I should do my PhD. And now here I am, 3 years into my PhD at Baylor. And it has been a very fun process, I must say. This is, a journey here was a journey in itself, but kind of all of this to say, you never know where you're gonna end up. And while you might have a great idea where you want to go, always be open to having new experiences, and always be open to trying something with research or classwork that maybe doesn't immediately spark your interest, but you never know what you might learn from it, because the experiences I had really did change where I ended up today. And I can say I'm pretty happy with where I've ended up today. And my favorite parts of grad school, like I mentioned before, this gets to be my office. And I wouldn't trade that for really anything. When I'm sitting at a desk a long time, I get a little antsy and anxious. So the fact that a lot of times my week means taking a boat out to a lake and spending time out there and traveling, it's just wonderful. Also being able to present my research at conferences. So I saw a lot of the posters you guys have done, which are extremely impressive, and I should take tips from you all because they're so good. But with graduate school, a lot of time you can travel to present your research. This was actually a conference at Victoria, Canada. So I was able to go to Canada for the first time, which isn't something I don't think I could have done that without graduate school. And then also my wonderful cohort that I have with groups like Present Your PhD that I'm here with now, they make it all worth it. So definitely, rely on your friends. They're very, very important. And before I kind of move away from it, every school is different and every department at a university is different. But the PhD process for Baylor Biology, I just kind of wanted to touch on so you have an idea of what it looks like. So typically it's 5 to 6 years for a PhD. If you have your master's, it can be a little bit shorter, so maybe 4 to 5, but this is typically around the time. A lot of this has to do with the fact that you're doing experiments, and experiments don't always work. And so then you have to find a different way to do it or redo it, get some data that you can actually publish and work with. You begin as a PhD student, and you go through a written and oral defense. You create a committee of 5 faculty members that you feel can help you with writing your research and going about everything. And 4 of them will write questions for you. You pass that, then you can actually write your research proposal and you present it kind of like I'm presenting this to you all. And then they kind of question you and decide if you can go on. Once you pass that process, you become a PhD candidate. So the 2 terms are different, and they're different for a reason. And then after you pass, then you do the research, you write the papers, then you have this final dissertation defense. I'm in my 3rd year, so right now I'm preparing for my research proposal, that last part to become a candidate, and it is 1 month from today. So yeah, wish me luck. Okay, so that's kind of enough about me and my path and how I ended up in front of you all talking about this today. So now I want to get into my research. So kind of the one big main question, why are lakes important? No matter what research you do, this is always something really important to start with. Your audience should know why they should care. I think lakes are an easy sell, right? Because we need water. But they provide many different services besides just drinking water, even though I would argue that that's the most important one. So lakes are a habitat for aquatic organisms, right there. But along with just being a habitat for aquatic organisms, we do eat fish, so that is necessary as far as a food source. But also recreation plays into this too. So recreation is a huge economic benefit to many, many states. So when you get your your fishing and hunting license right there, that goes into your state's economy. Whenever really terrible blooms happen, people can't utilize the lake, and so people aren't paying for those passes. They're not paying to go camp. They're not paying to utilize state parks that really rely on lakes and their lakes being beautiful. They're also good for hydroelectric power. So that's something within itself there. And then, of course, drinking water. So we get all our drinking water from lakes and lake sources. And whenever issues like algae blooms or even droughts happen, that really throws things for a loop there. Another reason freshwater lakes are really important is when we start thinking about where is our water, right? So if we think about total global water, oceans take up about 97%. We know oceans are saline. We can't drink that, at least not yet. The fresh water only really constitutes 2.5% of our total water source. When you break that down, most of that's in glaciers. And when we think about climate change issues and global warming, when those glaciers melt, that's going directly in the ocean usually. Well, that's more salty water that we can't access. So as far as surface and real fresh water that we can access, that's only 1.2% of the 2.5%. And then you break that down, and most of it's in ground ice or this permafrost ice. And all this to say, accessible fresh water we can utilize on this planet is only 0.003% of our total water. That's a very, very small number. And I think it's something that we take for granted because at least in America, most of us are probably able just to turn on a faucet or go to a drinking fountain out here and the water's there, it's drinkable, it's ready. A lot of other countries don't even have that. So keeping our lakes clean and healthy, important, very important. And one thing that really hurts our lakes is this term eutrophication. So right here you see two lakes that are suffering from severe algal blooms, and I would call this a eutrophic lake because what this term really means is a decrease in water quality, or if you break it down, I would say a well-fed lake, breaking the word down. And how eutrophication happens is a multitude of things are acting, with each other, but the main one being nitrogen and phosphorus, these two elements, but I refer to them as nutrients for algae growth, being put into our lake. With increased temperature, this only makes it worse. There's a lot of issues here with eutrophication beyond it just looking bad. So they do create anoxic conditions, which means there's no oxygen available in the lake when a really bad bloom happens. So aquatic organisms like fish, they require dissolved oxygen to survive. If there is no dissolved oxygen, we have big fish kills. And I'm sure many of you have probably seen pictures of it, or if you even go to the lakes, you might see some dead fish on the shore. If you also see an algal bloom, there's a good reason that might have been the cause for it. A decrease in water clarity. The importance with this, whenever that bloom happens on the top surface, light can no longer get down into the lake. So the green algae or the good algae that aren't really harmful or cause these blooms, they can't photosynthesize, so then they cannot grow and outcompete the kind of harmful algae I'll be talking about later. Decrease in recreational value of the lake. Like I mentioned, when people can't utilize the lake, money isn't coming into the state. And this production of toxins by cyanobacteria. So I've been using the term harmful algal blooms and blue-green algae. Really, the blue-green algae are not algae, they're bacteria. They're prokaryotes, they don't have a nucleus, they do a lot of things algae can't. So typical terminology, at least with news headlines, is blue-green algae, but from now on I'm gonna refer to them as cyanobacteria because that is what they are. They're bacteria that can photosynthesize. Like I mentioned with the decrease in oxygen, this causes dead zones. So the Gulf of Mexico is actually currently experiencing a very, very bad dead zone because the Mississippi River runs into the Gulf of Mexico, and almost all of our lakes and runoff run into the Mississippi River. So all these nutrients, all these pollutants end up in the Gulf of Mexico, which is a huge problem. So kind of the way a dead zone is created is this excess nutrient runoff happens in a body of water. You have a huge algal bloom because they have all these nutrients to survive. They're super happy. Eventually they do die. When they die, they float to the bottom and bacteria decompose them, but bacteria require oxygen to decompose. So they're taking all that oxygen out of the lake, and that's how we end up with what we call a dead zone, where no dissolved oxygen exists, so any aquatic life that lives there either dies or they have to move on. They have to either go, up or down the stream, whatever, to get away from that area and in a place where they can breathe, essentially. So as I mentioned, runoff is a huge, huge, huge component of eutrophication. Nitrogen and phosphorus are considered the biggest nutrient causes of eutrophication because they are the two most important macronutrients for algae to grow. They have a lot to do with like nucleic acids and metabolic processes, so they're very, very important. And also when I use the term runoff and watershed, anytime like a rain event happens, wherever that drains into the lake, that area is considered the watershed. And here we go, watersheds. So they do play a really crucial role in nutrient runoff because depending on what your watershed looks like, depends on what nutrients are actually available and can get into your body of water. Like I mentioned with my Oklahoma master's research, different nutrients are going to be able to access the lake depending on what is around it. And I think that, I'm going to skip to this one first. I think that's really important to think about when we think about development as well. So what this diagram kind of shows is when you think about a wooded area or a meadowed area, there's no impervious surface. So there is soil, there is like a catchment area where if a rain event happens and say a farm over-fertilized, so now there's a lot of excess nitrogen and phosphorus from the manure or fertilizer, it rains, it starts washing it down. If there's wooded area around that lake, that's able to catch that nitrogen and phosphorus before it enters the body of water. So it can stop it, and it's important. But the more we develop and the more we take that away, the more harmful these nutrients are to our lake systems because they can access them much, much, much more easier. So as far as residential and agricultural, if you had 3 inches of rain, maybe 2 inches will infiltrate, but a whole other inch will end up as runoff, and then it only increases from there. And watershed influences. Nutrient runoff isn't the only cause, but it is the largest cause. So that is why I focus on nitrogen and phosphorus exclusively. But air pollution, wastewater treatment, And then septic issues can also end up in our water. And these are just more pictures of lakes that have experienced this as an aerial view. These first two lakes right here, I'm going to talk about as far as my PhD research in a second, but these are from Lake Okeechobee, which is a lake in Florida. You might have heard that name in the news possibly, but it is experiencing a huge, huge bloom. And then over here is Lake Erie. So Lake Erie is very known for having this harmful algal bloom from a cyanobacteria referred to as microcystis. And this is really, really important because the Great Lakes constitute a ton of water for North America. So when this water goes bad, cities like Toledo and Ohio and other cities, they have huge issues because their water treatment plants aren't always equipped to handle these kind of problems. So the specific cyanobacteria that I study and that usually cause these issues, there's two that are kind of the big ones, and they're very different physiologically. So the picture I have here are two different species of the first cyanobacteria called microcystis, which produces microcystin, which is a cyanotoxin. So very, very similar names. It actually acts as a hepatotoxin, meaning it affects your liver. So if an animal were to drink a bunch of water that has been polluted and caused microcystis to grow, there's a good chance they'd have liver failure. If a human consumed a ton of that as well, same thing. And then Delicospermum is actually the other one. So it's the one that almost looks like a chain of pearls down here. And it produces anotoxin, which is a neurotoxin. And that's actually the thing that's been killing the dogs. So the bloom that happened down in Austin, it was an anotoxin that killed the dogs, and happened in other areas too. Anotoxin used to actually be referred to as a very fast death factor whenever it was first discovered, I think around probably the 50s, because since it is a neurotoxin, if you ingest too much, it immediately affects you, and usually animals will die. No human deaths have been recorded yet from this one, and I think that's one thing about government and things like that is they kind of wait until something bad happens to make a law or make regulation. So right now there is no regulation on how much anotoxin can be in our drinking water. That's a problem. Also, a lot of water quality places, whenever they test water samples, they do look for these cyanobacteria to make sure, okay, what kind of phytoplankton are making up the community? Do these exist? Should we be worried? But they don't usually test for toxins until something has happened, which once again is a problem. So like I mentioned though, these two are very different physiologically. So the Delicospermum here, it is filamentous. So all these individual cells combine together to create a filament. And it has a couple evolutionary advantages that keep it alive and help it outcompete green algae, or kind of the algae that can't produce anything harmful. One being that it is something called a diazotroph. And what that means is it can take atmospheric N2 gas and it can fix it within its cells to create ammonium. And that's really powerful because pretty much if we took away the nitrogen from its source, it can make it itself, and then it will continue growing. So it's hard to control for this certain cyanobacteria. It also produces resting cells referred to as akinetes. So the akinete is right here, kind of the larger one with the heterocyst being what they use to fix gas, being the clear one here. The akinete as a resting cell, whenever it dies, if it does die, that cell goes into the sediment and it can survive for, I would be willing to say, hundreds of years. And then when conditions are good again, it comes back to life. So it's very hard to control for these things because of these evolutionary advantages that it has. Microcystis is different. It's actually unicellular, but it exists in a colony instead of a filament. It is a non-diazotroph, so it cannot fix or create its own usable nitrogen. And there's no resting cells. So it doesn't have these same evolutionary advantages that this one does. So kind of what I want to find out regarding both of these is how do different environmental conditions affect their growth and toxin production? Mainly by the use of nitrogen and phosphorus stoichiometry. So what stoichiometry is just ratios. So if you think about a chemical equation, you have to have so many reactants to create a product, right? You can't make a cake with 3 eggs if you need 4. That's kind of the way I try to think about it. I also have a very typical diagram here of a barrel with many planks. The water represents yield. So if you're thinking about growth, you can only hold as much water as your lowest plank. So you can only have as much growth or yield as your most limiting nutrient or limiting environmental factor. So I'm curious in what ratio nitrogen and phosphorus need to be to limit the growth of these organisms, to try to figure out if that's a way that I can control for them. Also, I'm interested in salinity, mainly because of that Lake Okeechobee that I mentioned that's in Florida. So with rising sea levels happening because of climate change, eventually more oceans and freshwater are going to start mixing. What will that salinity do to these organisms that are not used to being exposed to that? So once again, this is Lake Okeechobee. And one management thing that Florida is currently doing right now when these blooms happen is they're managing it by mitigating it. They are pumping this water into the ocean. So that's how they're getting rid of the bloom, is, okay, let's just move it. But we don't know what's going to happen whenever this harmful algal bloom hits ocean conditions, right? So that is what my work is kind of looking at right now. So the cyanotoxins I've mentioned, I kind of just want to show a really quick molecular figure of these. This is anatoxin A, that's that neurotoxin. And then that is microcystin, that's the hepatotoxin. Microcystin is a much bigger molecule, so it's harder for it to break down. It's also very nitrogen rich compared to anatoxin A. So knowing this and just even seeing this molecular structure difference, it made me kind of wonder, okay, so when microcystis is exposed to a lot of nitrogen, it has to make choices. It has to make some trade-off decisions. Where will it allocate the nitrogen? How much of it is it really putting towards this harmful toxin? And the way I decided to try to figure that out was by growing up more bioassays, kind of like in that picture I showed you before. So there's another diagram of what picture of what microcystis looks like. I put them in beakers of a nitrogen to phosphorus ratio of 4, which is very, very low nitrogen, and a nitrogen to phosphorus ratio of 50, which is really high nitrogen. So you can think about this as for every unit of phosphorus, there's 4 units or molecules of nitrogen. And then here for every one like atom or unit of phosphorus, there's going to be 50 nitrogen. I grew them up for 21 days because I knew around that time the very low nitrogen condition would become limited. And then I spiked them with salt, which ranged in salinities from 0 to 10.5 grams per liter. And I sampled on days 1, 2, 4, 6, and 9 after that for growth and then toxin production. Visually, this is what my cultures ended up looking like. So right here, the ones that are almost like the pale yellow, those are my very low nitrogen conditions. And regardless if it was the control or the very high salt, the cells were pretty much already dead. They didn't have enough nitrogen to continue growing anyway, so adding the salt didn't really visually change them. Now the one that had high nitrogen, this is the control, so no salt was added, but you can see how much greener it is. So it was in a much happier condition. It could keep growing. And this is what happened whenever I added salt. So salt is dense, so I didn't expect there to be some kind of physiological change, but I had never seen this happen before in my cultures. Usually with microcystis, it looks just like it did previously. It's all homogeneous throughout the jar, but this, they started clumping. And to me, in these high salt conditions where they clumped, that is another, what I think, evolutionary advantage. They are trying to avoid the salt. Right? Because we know with like hypo and hypertonic solutions, when salt, comes in contact with cells, they may lyse or they may shrink. So I believe the microcystis here is trying to avoid that the best way they knew how. Another great advantage to cyanobacteria is they have gas vesicles. So they can, if they want to be higher up in the top of the water, they can be. If they want to be lower, they can be. They can move as they wish. I think what they were doing here was clumping together, so the salt was infiltrating the outer cells and the ones on the inside were protected. Another trait just to continue survival. This one was hit with around the middle amount of salt, so you can see it's still trying to avoid that bottom area where that density of salt would have happened, but it's not as insanely, clumped like this guy was. And the data I was able to get from this was just as interesting as the visual kind of led me to believe. So in panel A, we have that low nitrogen growth condition. And what I'm showing you is the term relative fluorescent units, which pretty much just measure growth. So throughout this experiment, I took them about every other day. And the red arrow indicates where I added salinity, where I added that salt. So the RFUs for the very low nitrogen conditions, they stalled around 1,500, I'd say. They got there, then they became limited, they could no longer grow, and they kind of just hung out there. And even after I added the salt, it didn't matter. Nothing really changed. With the high nitrogen condition, their growth was able to continue growing, even up to the point where I added salt. And salt concentrations of less than 5.6 grams per liter weren't really negatively affected as far as growth. They could keep growing. However, once you kind of hit that 5.6 and 10.5 grams per liter, they were negatively affected. And you see that really nice zipper effect that happened where they immediately started decreasing. They were dying. And as far as the actual microcystin that I was interested in, so how did this affect that cyanotoxin that is the harmful part for us? Panel A once again shows low nitrogen, panel B shows high. So remember, it was a very nitrogen-rich molecule, so I did expect the more nitrogen in its environment it had, the more it could allocate to this toxin, and that's what I found. So as far as the low nitrogen, very, very little was actually found to be extracellular or dissolved in the media. But as far as the high nitrogen goes, over the course of those 9 days I tested after salt addition, there was a huge spike in the 5.6 grams per liter dissolved right here. And to me, what this really means is whenever these things hit the ocean, if they were to immediately go into the ocean, there still would be a little bit of dissolved microcystin. But there are things called estuaries, which are a mix of freshwater and saltwater, so not as salty as the ocean. I think that's where this would really, really hurt those areas. This also just kind of shows that my hypothesis about the more nitrogen, the more microcystin was correct. So on the x-axis, I have particulate nitrogen, which is how much nitrogen was intercellular, so how much was inside the cell, and then total microcystin, so microcystin inside the cell and outside the cell. And there's a really, really strong, good correlation here that shows the more nitrogen it has, the more it can allocate, and it is allocating it to this. So kind of I came to two main conclusions from my research, which is high nutrient conditions do promote cyanobacterial growth. And when microcystis specifically is grown under a very high nitrogen condition, more microcystin will be produced and it will be released under salt concentrations of greater than 5.6 grams per liter. I think that this is a really important conclusion because this tells me we really need to control for nitrogen. So in agricultural, use of fertilizers, a lot of times they over-fertilize, and I think that's something agricultural research is probably going towards, is how can we still get very good crop production but not use as much nitrogen that can end up in our water and then end up producing cyanotoxins like this that could be harmful for us and other animals. Okay, and with that, that is my talk. I really appreciate you guys being here and listening. And I have both my Twitter handle as well as Present Your PhD if you'd like to learn more about this kind of research. So thank you.
Well, we appreciate you coming out.
Yeah, of course. Yeah. Any questions or is it?
Yeah, you can ask questions. Anybody got any questions?
Sure, yes.
Are there any freshwater lakes, places in the United States where algae does not develop?
So I would say really up in the northern part. I would not say they don't develop at all, but I would say they have a much harder time blooming. They'd have a hard time becoming so, intensely large that it's an issue because of how cold it gets up in those northern lakes. And a lot of the northern lakes are, lakes that were created by glaciers. So their watershed isn't really agricultural. So there's not a lot of nutrients going into it, and it's also very, very low temperature as well. Mm-hmm.
Any other questions?
All right.
Okay.
Thank you, guys.